Good. Hello, everyone. My name is Chris. You can also just call me Chris. I will give 5 lectures, essentially on phase separation, biomolecular condensates, and I will start each talk with a few slides, just to get you started, to think about what is exciting in terms of ongoing science. But then, in particular, the first lecture should bring everyone on the same page. For some, it might be a little bit of a repetition, so we'll start with an Ising type kind of model, derive a free energy, and discuss step by step the involved physics of phase separation. Yeah? I will always take, even for the experts, let's say PhDs or senior master students, I will also give some extra angle that you definitely hear something new. However, yeah, let me maybe get first started. And please interrupt me and ask questions. I mean, the lecture is for you guys in case for sure I have too much material with me, so I will not be able to finish anyways. So you can slow me down at the right moments, and then I explain more detail. I'm happy to do this. Yeah, because the lecture is for you guys and not for me. Good. Let's start with why the physics of phase separation. I mean, it's an old discipline, actually. There was a peak, I think, in the 70s and also the 90s in polymer physics, thinking about phase separation. And there was essentially the goal to avoid phase separation. And now people have found these very exciting things in cells they call biomolecular condensates, and they're very abundant. There have been tens of years of explorations of hundreds of examples in different cell types, at different stages, and different stress conditions, and they were just overlooked. And because when you, I don't know how much biology you had in your courses, but I think this is something I would assume you know. The cell is highly organized, and it's highly organized by essentially membranes. And this we have seen already since we have a light microscope, because you can look into a cell and you see them in the light microscope. You don't need a fancy modern microscope for that. And you see all these membrane-bound organelles. Here's the nucleus, which hosts the genetic material, mitochondria, Golgi apparatus. Here are the mitochondria, these little things. Actually, there are extremely many. And they obviously spatially organize it, and they have a membrane, a double lipid bilayer with embedded proteins that creates a barrier. And it's amazing to keep certain biochemical and physical properties different between the inside and the outside. That's what it was made. It was made to be a barrier, but you have to think about transport, because the barrier, I mean, yeah, it's a doorkeeper, so you have to then invent channels to let certain molecules in and out. There are other ways how the cells are spatially organized, and this is by also by these condensates called biomolecular condensates. And at the beginning, they were called membrane-less organelles. To make this point, they don't have a membrane. And for some people, it was quite a surprise that can be stable for so long time, and they can contribute to the dynamics of cells so interestingly. But from a physics angle or polymer physics angle, this was no surprise, because if you have a mixture of long-chain polymers that they phase separate is kind of the expected scenario. Yeah? And obviously the cell in a serious order approximation is a bag of extremely many complex polymers. Yeah? Good. So here are a few examples. It's not complete. So if you look on Google Scholar, every day there are about 20 new publications in that field, or even more. It's just crazy. I can't catch up. So I will not even try to be complete, even up to date. So I even started here with the really original references of the first observations for these different kind of condensates. And what you see here are simply different parts of the cell. This is inside the nucleus, so even at this very small scale, so inside here, you have droplets. Here's an example I will have through the entire lecture, because I worked on this also as a scientist. This is a model organism, the C. elegans embryo. This is an eggshell, so it's one cell stage. Here it has 2 nuclei, it's just divided, and it actually has these little dots, which you see here. They're called the P granules. And I will talk about the P granules in more detail, but these are essentially RNA protein condensates that are liquid-like. Yeah? And there are many other examples. And so if you want to draw a new sketch for textbooks, you can please make it nicer than this one. You would then say, there are membrane-bound ones, but also many that are non-membrane-bound. Yeah? And this interplay kind of is very important to understand when you think about spatial organization of the cell. Good. So let me show you one movie that needs a bit of explanation, because what you see here, this is a typical experimental scenario for confocal microscopy. You see a protein. In that case, its name doesn't matter. It's PGL. Doesn't matter what it's doing. It's labeled. It carries a label and starts to send a light through the microscope, a fluorescently labeled, a GFP. And that's why you see these little white domains here, yeah, okay? But you don't see all the rest because you haven't labeled it. That doesn't mean it's empty, okay? That's why I have added here a little bit of a helper, because particularly this beamer is a bit brownish or blurred. Or is it my glasses? I don't know. No, it's a beamer. You see, here is actually the nucleus. So this is membrane-bound organelle, and these objects here are called P granules. So these are exactly biomolecular condensates as you would see them in the microscope. Quite blurred because of optic effects. But what you see already is the first interesting observation you should make is they have wetted a surface, the membrane-bound organelle. And wetting is a property we know from droplets. Yeah? Now let the movie run, and what you will see is that they drift off, deform, fuse, and relax back to a spherical shape in one movie. And that's why I like this movie, because it shows all the important properties we know from liquid droplets: wetting, fusion, shear deformation, and relaxation to a spherical shape, which indicates there's also a surface tension. When you put this into numbers for this, not for that specific case, but in general for most biomolecular condensates, you can put physical parameters on this. And let's start first with the length scales here. The length scales, how big are they? Sorry, here the scale bar has vanished. Their size is typically, the nucleus we have a few hundred nanometers, and the P granules are very big, so they have a few micrometers. So that's the scale we talk about. I mean, a cell depends on cell type, we talk about a few micrometers up to 20, 30 micrometer for this embryonic cell. Then, what's the time to grow? Okay, the time to grow, well, you see them. This was a live movie. You see here even the label here. This kinetics was on 60 seconds, so it's a minute. The typical timescale for nucleation, growth, etc., is in the order of seconds a minute. So this is quite, you can literally monitor it live. You don't have to re-render the movie. It's quite cool. And you can even estimate by various techniques. I will not go into those details, but there's a project on that. So stay tuned if you want to solve that. I mean, how to use theory to extract some of those physical parameters. And one important one is how fast molecules move. Because they're liquid, there's diffusion of the proteins. And the diffusivities are rather high. So they're in that, some they're a bit lower, some a bit higher. So what you should know is kind of if you've never seen these numbers, so if you put just a small protein, nanometer big, in water, typical diffusivities are 100 micrometers squared per second, or 80. GFP is a good example. Okay, this is a good reference. You see, like, oh, in the cell, a bit slower. Depends where you diffuse. When you diffuse inside, you also get even slower, but it's quite high. Yeah, you just have to wait a little bit longer until you see the molecules moving around. So they move quite actively. And I think when Patricia gives her talk, I think she will talk about membranes, and also you will see the rather similar diffusivities also in the membrane in the order of a micrometer squared per second or lower. So it's quite liquid. Viscosities, I don't know whether you know the viscosity of water, but we should consider to be a low viscosity liquid. So when you measure this, of course, depends on the different kind of stages. You get a lower viscosity than water. Yeah? So speak in terms of olive oil, I think everyone uses olive oil from time to time. Depends where you buy it, whether it's old or new or mixed, that quality. That's also the range typically of olive oil. Yeah? So that's how you could picture them, how they flow. They flow like liquids in the kitchen. Yeah? Good. So it's a liquid. So what is it good for? It's good for many things, and this is exactly ongoing research. And I will only show you this sketch. It's an outdated review article, 7 years old, but it was extremely predictive in terms of the type of functionalities condensates can have, either activating chemical processes, forming filaments, buffer concentration fluctuation, creating forces. There are a large body of works that has emerged, and I will not go into these examples in detail, but here you see some nice references of selected papers that explain a little bit what condensates can do in living cells. But to be fair, the community has also a bit of a problem because it's a huge expectation. So many people jumped into the field. So the number of new discoveries is limited compared to the amount of researchers in that field. So now this field definitely needs a breakthrough in terms of how this really changes our fundamental view how cells organize themselves. Okay, this is I can skip. Good. So I show now one thing very shortly. what we did, because we felt like, yeah, wait, it's if they're liquid, it should behave as a liquid as we know it, as a polymer liquid. And I don't know, because now I'm in Brazil, I don't know, do you know what ouzo is? No, it's not the thing, yeah, okay. So at least I'm sure you have an analog. Well, let me find the analog. So in the Mediterranean Sea, I mean, each country, Greece, France, Italy, etc., have all their anise-based, anise is an anise oil, it's an oil, based liquor. They're called either raki, pastis, ouzo, etc. And it's a mixture of water, alcohol, obviously, 40%, yeah, for the fun, and anise oil for the taste, a few percent. And with the alcohol, anise oil is fine. And if you do this, is actually an experiment I did. You can warming up. This was not a good idea. Okay, I warmed it up. Don't do this with liquors. So before, when you put in the ouzo, add water, it becomes cloudy. And that's an observation we will understand today. And this is actually because you have little droplets, anise oil droplets, that don't like the water. They phase separate, and they scatter the light. It's not critical polysins. It's something else. It's really the scattering of light because we have here little droplets in the range of hundreds or tens and hundreds of nanometers and bigger that scatter the light. Good, but if you warm it up, what happens? Yeah, you push entropy, right? And entropy wants to homogenize. So no spatial organization possible, everything gets disordered, and it gets transparent, the light goes through. Yeah? And if that is true, okay, that is true, because I did the experiment, but you can also do this in the cell. You can do this in the C. elegans embryo, a living cell. Don't do this in a human cell. And we can sustain 2 degrees Celsius and then we start dying. Yeah? This is actually, this creature is fine. I'll tell you in a second why, but let's do it first. So what you see here is exactly this confocal microscopy. You see here these P granules, these apparent liquid droplets. And then there will be two lines now. So actually we measure the total condensed volume of those. So we just add up all those volumes. So we segment the image. So we go through 3 dimensions to the entire image and calculate the entire volume. And we also then change the temperature. We put it into a heat bath and go up with the temperature and down. And now watch what happens. Always when the temperature is high, they dissolve. When the temperature is low, they form again and grow. And you can do this many, many times. And if you then let it sit, it divides, a worm develops, it hatches nicely, and the creature lives nicely. So it was fine with that, nice experiment. Of course, it was more a physics type of experiment to test how well they're really liquids and how much reversible all this kind of formation of these biomolecular condensates is. Good. Why can we do this? Because it's a cold-blooded animal. And it's completely used to, it lives in the sand of beaches all over the world, C. elegans embryo, and it's used to live with temperatures in the range of 5 to 30 degrees. Above 30, it gets stressed. Maybe that's why it doesn't live here in Brazil, maybe likely, but I have to look it up. But at least in Europe, you have it in all the beaches because they're much colder than here. Good. And then you can see actually at low temperatures, you have a lot of these P granules, and at high temperatures, they dissolve. Okay, what you can do is you can calculate a phase diagram for that, and this is exactly what I would like to do today. I would like to derive how to calculate this phase diagram today. And this phase diagram, we actually measured it in the cell. So for each temperature, we measured the concentration inside and outside over extremely many P granules. And what we found is exactly this phase diagram. This is extrapolated just to indicate the shape of here this dense branch, what's the concentration inside, and here the concentration outside. And the model that we used is exactly this super simple, what's called Floer-Huggins free energy density, which is exactly comprised of a mixing entropy with a temperature-dependent molecular volume and an energy interaction term. And who has seen that free energy? Please be honest, because then I know how fast I have to do the calculations or not. Okay, this is good, so you will not get bored, it will still be interesting angle. So it's good because we derive it. Though you haven't seen it, that's great, so we derive it together, so it will not jump out of the blue. Okay, what we can do is, what we did is there are 2 ways. We can either recast the data in terms of these 2 temperature-dependent parameters, chi and nu, and plot them, yeah? And then fit this line, yeah? This is what we did here. We fitted this one, here this is this dashed line, we fitted that one, and then calculate the phase diagram. And what you can see here, the dashed line agrees pretty well with the data. And you can also globally fit this phase diagram and see, that's a solid line here, how well these parameters agree. So it's kind of 2 ways of fitting, super consistent. Critical point here at the top, this is called the critical point. Who knows what's the critical point? Yeah, okay, that's good. Okay, but we come back to this one, what is happening at that critical point. This critical point is almost very similar, but it's hard to reach anyways because the organism dies at these temperatures. Good. So last slide, and then I go to the chalkboard, and from that on, I just keep on calculating. Why is it relevant? Why do we care as physicists? And this is because, of course, when we describe such a complicated organism like in the cell, it's not clear in what respect we can use fundamental principles as we learned in our courses, for example, thermodynamics, because the cell is always an out-of-equilibrium system. But it doesn't mean that thermodynamics doesn't govern the behavior of the cell. With thermodynamics, I do not mean equilibrium thermodynamics. Okay? Why is thermodynamics so beautiful? Because, yeah, it's a closed theory, but we can rescue it maybe locally. So that we can construct local elements where local thermodynamics governs the system, and the entire out-of-equilibrium dynamics is set how these volume elements talk to each other in space. And this is called non-equilibrium thermodynamics. But we have to, this relies on the idea that we can think about local volume elements that are at local equilibrium. And if you can't do this, we have to come up with a super generic type of theory. Yeah? And if not, we can rely on those things and move on and make much more precise predictions with much less parameters. And actually, this is the case, and we can show this even here for the C. elegans embryo, and this is very easy because we have to look at the length scales. I mentioned already the P granules are micrometer sized. This is such an embryonic cell. This like 50 micrometers to get a flavor. They form on seconds and minutes. So what are the out-of-equilibrium features here? The first one is we maintained it out of equilibrium. So we changed the temperature. Changing the temperature means, yeah, you bring it to a new state, there will be some dynamics. So you have to solve the heat equation. You can do this, and heat transport, do you think it's fast, slow, compared to diffusion of molecules? Diffusivity of heat? What is diffusion of heat? What is it? How can I think about this? Vibration of water molecules is super fast. So just water, the cell is full of water, first of all. It's 70% water. So the heat transport is almost the same as for classical water. It's very fast. So if you let it go, you can calculate it takes about a few tenths microseconds for when you go from, let's say, 10 degrees Celsius to 20 degrees Celsius to relax the profile. Okay, this is not the thing, but the cell is an out-of-equilibrium system. It produces heat. Why does it produce heat? Does it have fever? No. Why does it produce heat? Yeah? Say again? Sorry, I could just not hear it. Sorry, say again. Yeah, the entropy, yeah, it's out of equilibrium, so there must be an entropy change, yeah, true. But where does it come from? So how does the cell produce heat? I mean, come on, we produce a lot of heat, right? That's the problem, huh? Why? Exactly. There are lots of biochemical reactions running. We're turning over high fuel molecules, really high fuel, about 10 kBt. Put this into a Boltzmann factor. This is an enormous amount of energy per molecular event that we burn, and you can actually calculate this down. And anyone who's want to get more to these energetics of cell, there's this beautiful lecture series, but also books from Rob Phillips. I can highly recommend to have a look. But when you calculate it, it's 1 watt per liter. It's rather conserved throughout the cell. It's a very good average. Maybe in the mitochondria it's a little bit increased. You can calculate this one. It will lead, of course, to a temperature gradient, right? Because you produce somewhere in a body, and what's happening outside? Outside sits the heat bath, and you fix the temperature. So there will be a temperature gradient from the inside to the outside. And you can calculate that one, and no one can measure that one. It's super little. 10 to the minus degrees Celsius. So this doesn't matter. Let's look at the single metabolic events. So burning a single ATP should lead to a temperature change. Yeah, you see lots of little point sources that always create heat, and they dissipate through the system. And you can estimate this, also not so difficult. You solve the heat equation with a point source. You see that? This is the amount of ATP, point source in time and space, and you ask in the end the question, how long does it take to go to that the temperature? So how much what time does it take, and how much does the temperature change after a single event in a certain volume element? And now I make the volume element bigger and bigger. And the surprising thing is, when you look at a very small scale, like a cubus of 1 nanometer times 1 nanometer times 1 nanometer. Okay, that's the size of. Why isn't 1 nanometer interesting for a cell? That's the size of a protein, typical size of proteins. Yeah? So that's exactly where the dissipation happens. The ATP is somehow hydrolyzed by an enzyme, for example, and there's a quite significant temperature change, 3 degrees Celsius locally. And this small volume element is enormous. It shocked us a little bit. That sounds like fever, but it doesn't, because it takes to relax. It relaxes very, very quickly. So even on a 10 nanometers cube length scale, we have already just a temperature, almost negligible, unmeasurable temperature change. Which means you have all these little events. They create the temperature change on a small scale, but they homogenize very quickly. Okay. So what I can think about, even though I have such a setting, an out-of-equilibrium setting, I can define a local volume element with a local temperature, but I have to think about the scale and I have to think about this volume element. And this is, we can discuss whether it's 10 nanometers cube, 100 nanometers cube, doesn't matter. But which means we can do local thermodynamics in that volume element. Okay, so we can run easing like kind of, we can calculate the partition function locally in that volume element and do our stuff and think about transport among volume elements. Okay, where does the out-of-equilibrium nature now? Okay, still an out-of-equilibrium system. Where is this out-of-equilibrium now going? It's gone, right? No, it's not gone. On a larger length scale, of course, system is out of equilibrium because there are reactions that come with a rate, and we discussed this also in the course, and the diffusivity, they make a reaction diffusion length scale, which is, you can estimate it for all kinds of system, is in the order of micrometers to hundreds of micrometers. They create all this out-of-equilibrium physics you know in the cell, all these patterns, Turing patterns, cytoskeleton organization, all this kind of stuff, and it happens all on that length scale. Yeah? So which means we can use local thermodynamics to describe these systems. Okay, wait, I only showed this for the peak granules, and of course now you have to, in principle, to show this for all condensates, and there might be condensates and symmetric condensates that violate this, yeah? But no one has looked at this. So we only looked at one system and showed it for that one, the peak granules in the C. elegans embryo. Now I would switch to the chalkboard and talk about thermodynamics, but I hope I gave you enough reason to think about, okay, thermodynamics is relevant for that topic. Let's start from the bottom up, and in the end, our goal is to end up with concepts describing biomolecular condensates away from equilibrium. Are there questions you want to ask me before I switch to calculate something on the board? Yeah, please.
So I simply wonder if the cell volume changes because you increase the temperature, right? I mean, in the experiment, like the previous video,
Yeah.
because the membrane should get more porous, and then if there is an influx, and
you talk about this movie?
Yeah, I mean, you do confocal. Like, I wonder actually if you have information 3D about that.
Okay. Yeah, okay. Okay, you're an expert. Awesome. So what we actually do, we do three-dimensional confocal. So we segment all the information in 3D. What I show you, just that you have a movie to see something, it's projected. So I project all the peak granules. So they actually live in 3D, so they in the end, much more sparse or dilute than it looks like because it's projected. Yeah. But we use the full 3D information to segment the image, to calculate concentration inside, outside, exactly. And that's consistent with the point spread function. But we can talk about this more in detail. This is exactly all in this PNS paper that I also we have seen this. We shared some material, right? References, and I shared this reference as well. It's all described there. This was that's why it was took 5 years this work.
Oh, hi. Looking at this graph, as the time goes, the peak is becoming higher and higher. There's some kind of memory in the cell, something like that?
Not that we know of, but something else happens. So this is, you see already in the movie, where I can go back a bit. There's a gradient forming in the system, lecture 2.
Okay.
Yeah? And this will describe as well. So you see this also at the very end, that it's no more that homogeneous. It's a bit of a bias. Let me see. It still looks homogeneous. Yeah, okay, maybe I cut it in the right way. But what happens exactly on that time scale, roughly around 20 minutes, a concentration gradient, max 5 protein gradient develops, and it actually segregates the P granules just to one side of the cell. Because this is not part of, the cell wants to divide asymmetrically, having only the P granules in one of the daughter cells and not in the other one. Yeah, but this is lecture 2, and we just show a movie for that. Yeah. Good. Yeah, then let's let's start then with the actual lecture. I want to start with something that you have all seen in your basic physics lectures. This is, of course, the Ising model. And the Ising model is a lattice model. And we will use this idea to derive this free energy to describe exactly this phenomenon. So we start with an Hamiltonian H of a binary mixture. Throughout the lecture, I will generalize it later to ternary and to multi-component mixtures, because we want to describe also chemical reactions out of equilibrium. We have a system with m lattice sides. So we can draw it here. And we put molecules there. Maybe have like colors. Okay, here are colors. And they need not to be, they can actually occupy more than one lattice side. Here this could be A, for example. And here there might be another A, okay? This occupies now two lattice sides. The lattice side, I mean, we can just generalize this to d dimensions. I mean, come on, but life is 3D. So this has, I can only draw in, of course, in 2D, okay? So this is the lattice size volume, and I call this nu. And of course, the lattice is fully, there are no vacancies. It's fully occupied by A and B particles, okay? Just A and B. I don't want to draw it everywhere now, but I think you get the point. It's just A and B particles, okay? We can define a molecular volume here. Actually, I can use this here. Yeah, I mean, it's a bit weird to call it molecular volumes. They're not yet molecules, it's just things on lattice, but I call this nu I. And write here Ni times nu. So there are multiples of the lattice size volume. And I can also define the system volume simply by Na so M lattice sides, exactly. Yeah. And what is Na and Nb? This is simply Na I write here as the particle numbers of A and B respectively. Good. And the total volume, of course, I can simply write as V, which is simply M times nu. Okay? Good. This is just the basics, just to have notations clear. What you can see here nicely is you can divide by M, yeah? And when you divide by M, you get a 1 here. And you get some sort of a fraction, and this I call, in that case you call it called particle fraction. But you can call it also, of course, volume fraction. Here in that case, doesn't matter. And the volume fraction of particle I is simply defined as nu I, Ni, divided by the volume. And you can see nicely, you can also write in terms of a particle fraction, but just dividing, taking out this nu here. Good, but later I will talk about volume, so it might be nicer to not introduce many different fractions. Good. So, on to statistical mechanics. So we have to think about a state variable. And of course, this system is, yeah, it's like the Ising model, but it just, instead of spin up and down, we have particles A and B. And the state variable I call, in addition to the Ising model, also sigma n. It can have two states. And the state A is, oh no, okay, the other way around. If the state variable is 0, then there's a B particle. If it's 1, then there is the A particle. We can then define a microstate. On the lattice, which is simply sigma 1. This is one lattice height I, 1, sigma 2, sigma m, because we have m lattice heights. And because we do discrete mechanics, we have to, in the end, calculate a partition function. So we have to go over all possible microstates. And the notation I use here is sigma. And right, also quite commonly used, we use these curly brackets to indicate all the possible combinations of arranging A and B particles here on the lattice. Okay? Good. So different to the Ising model is we have these two different particles. Oh wait, I have a color. We can think about different interactions, BB interactions and AA interactions. This we don't have for the Ising model. And for that, we introduce parameters, interaction parameters, and I call this epsilon AV, or E. No, I call it E, not epsilon, E. EAA and EBV. Okay, these are my parameters. And in some way, when you think about real molecules, this comes somehow from this dip and also the repulsive part of this Lennard-Jones potential when you integrate out a more molecular type of theory. So what does it mean? So if E ij is positive, and I can have E ij negative. So it's positive, we follow exactly the same classical convention. If it's positive, I doesn't like J, so it's repulsive. And if it's negative, it's attractive. Good. So this is the model we want to start with, similar to the Ising model, but we will now find a few things will be very different. What is the aim? The first aim is, I hear myself breathing, it's a bit weird, for you guys in particular. We want to calculate the free energy. And I have to choose an ensemble. The ensemble I choose is the canonical ensemble with the Helmholtz free energy F, which depends on temperature, volume, and the particle number, Na. Why just Na? Why not Nb? Because if I know where all the Nas are, I know where the Nbs are, where the Bs are. Okay? That's very important. So we have no freedom here to write here in on the lattice Na and Nb. And the free energy, of course, this you all know, I can calculate from the logarithm of the partition function. And the partition function is simply, in the canonical ensemble, the sum over all possible configurations, capital omega, weighted by the Boltzmann factor. And for that, we need the Hamiltonian relative to KBT. Good. In this ensemble, we have to, what's very important is the differential of the state function. It's classic as for classical gas, it's also minus S dt. Then, and this is usually what is not taught in classical thermodynamics lecture, there comes a new thing here that is not the pressure P, it's the osmotic pressure. Yeah. And we also get the exchange chemical potential. These are two things that people are not used to, and this is usually it's the osmotic pressure and the exchange chemical potential. What are they and why do we need those things here? So first of all, let's define here the important derivatives. The entropy, even if we don't use it, is simply the free energy derivative with respect to temperature at fixed volume and fixed NA. This is the entropy. The osmotic pressure is minus the entropy, the derivative with respect to volume at fixed temperature and NA. And the exchange chemical potential a very important object for the entire lecture is mu bar is simply the derivative of the free energy with respect to the particle number at fixed temperature and volume. Okay? So, short explanation, of course you can derive this. Of course you can start with the classics, the two chemical potentials here and the pressure, and actually you can derive this. And it comes exactly from this constraint I mentioned at the beginning. Yeah? When you know A, B is fixed. And why is this? Because, where did I write it? Have I written this? Yeah, exactly. It's because of this one here. Yeah, because if you give me an A and an B, the volume is determined. So I cannot perform a derivative back to NA and keep NB and the volume fixed. I can't do this. And this is exactly the choice of the ensemble where the volume is fixed. So I have to lift this constraint and allow myself to only change NA at fixed volume. And this is exactly where then this exchange comes from. The exchange chemical potential and also the osmotic pressure can be considered as an exchange quantity. Good. Yeah, so that's because later I showed you this free energy density. Yeah? There's this small f. We want to rewrite this derivative with respect to the free energy density, and we can just plug this in. So you see here f, we just plug this in, and we can actually, we don't have to worry because here we have to be careful because we have the derivative acts on v and f and on v. Yeah. So if you do this, so what do we get? We get minus. So if first the derivative acts on this one here, I get minus f. Then what else do I get? Then I just leave here the volume and write df respect to dv at fixed t. Yeah. And this I can simply massage to a volume fraction. I should have written this right away. I write it here maybe. Minus f plus f df respect to d phi. Yeah. Good. That's the same I can do for the exchange chemical potential. So if I plug this in, it's at fixed volume. So I can divide here by the volume and here create in the end a density and rewrite this in terms of nu df over d phi at fixed temperature. And why is this powerful? Because we will derive from coarse-graining free energy densities. Yeah? And now with these free energy densities, we can calculate these intensive conjugate variables to the volume and the particle number. Good. All right. What is missing is the Hamiltonian. The Hamiltonian, I write it here now, and we can now specify it with the help of these interaction parameters. So I will use, as in the Ising model, next neighbor interactions, which I write here NN. Nearest. Nearest neighbor, NN. And I would like to first start with the interaction between AA. And this I can now nicely do because A, here, when I write sigma n times sigma n, this is only non-zero if it's occupied by an A. Yeah? So I can write here sigma n, sigma m. Yeah? So you see, like, it only contributes if there sits an A where the state operator is 1. Analogously, I can also do it for the other interactions. Here for the B, it's simply 1 minus. And for the interaction between A and B, we write a symmetric way. We wrong side. Sigma n 1 minus sigma m plus 1 minus sigma m sigma m. Okay, so you see this is the sum of all this. This is the Hamiltonian we look at. Yeah, it has the same structure as the Ising model. Yeah, essentially it has the complexity of a sigma square interaction, but it comes with these interesting extra parameters where we can distinguish whether A particles interact. Where did I write it here? A interacts with B, A with A, or B with B. So we can, without much effort, rewrite this a little bit. This is not just rearranging terms, yeah, just algebra. So I have still here 1/2. The 1/2 I have because of double counting. So I can rearrange this and have a very nice form and have here actually 2. Or I write here immediately, I can define a new parameter called delta E. Sigma n 1 minus sigma m. Okay, you see, like, this is kind of a squared interaction with a new parameter. I define it, I write it down in a second. And then one can't move the board, yeah? And then I get here linear terms. Okay. And this parameter is important for the lecture. It's an interaction parameter. Delta E. And it's simply the difference between the AB interaction it's a very simple thing, and that will pop up in our free energy after we had coarse-grained it. Yeah. The linear terms are actually not so relevant. And I think you know this from the Ising model. They just give a paramagnetic contribution. And actually, yeah, so the core thing is exactly in this sigma n times 1 minus sigma m. So, how to solve this? How did you solve this in your statistical physics lecture? What did you do? What did you do? Can you solve that? Has anyone ever solved that? Onsager solved it for today. Yeah? But what did you do? In the
a basic mean field approximation.
Yeah, exactly. So mean field. You can do mean field. And what you usually do is, so mean field, you replace the local interaction with the mean interaction, but with the same mean interaction at all the sites. And this we can't do. We could do, but we will miss a term and regret this step a lot later. So I will sketch you now the procedure how to do it for the phase separation problem. As a quick thing, that is hard to solve. because of this nonlinear interaction. And why it is even harder, because the equilibrium state we are aiming at, what is the equilibrium state in the Ising model? If something interesting happens, either so, yeah? All spins up, all spins down. That's a homogeneous state. That's the equilibrium state. So that's why also global mean field, I call this global mean field. What you do classically in the lecture is absolutely fine. But our equilibrium state is heterogeneous. That is actually the difference to the Ising model, because we have a conservation law that fixes the amount of A and B particles on the lattice, both. The spins are not fixed, they can flip, right? So it's like the conversion, so all become A, all become B. But now the A's and the B's, they can only exchange sites. So the state has to be heterogeneous. And how it looks like, well, the state operator can be 0 and 1. When you do a continuum limit, which you usually do when you do coarse graining, you get some field, I call it phi, which is a phi of x. This was between 0 and 1, so this doesn't change. So this will be now between the continuous range, 0 and 1. But the state at equilibrium that we aim at looks more like this. And what you have here, this thing is called an interface. So if you do a global, so if you think about this, when you go back to the lattice is fine-grained. What does it mean that you always replace interaction with the mean interaction everywhere? What does that mean? Yeah? Can't be true. No? And the problem is I cannot do the full calculations because it's a 5-6 hours lecture. But I can sketch you the steps, and the people who are interested, they can have my notes, and I made big boxes with the calculations, and you can really go through all these calculations in detail. But I promise you, with this recipe, you could do it yourself in principle, okay? But please ask questions, because there are methods involved. There are really beautiful concepts that I would like to highlight, and please ask if you have not seen that. So what's the procedure? So the goal is, of course, to get the partition function. If you have the partition function, of course, we can calculate where is it. We can calculate the free energy. So this is what we want. So let us start with the Legendre transform of the free energy. In the canonical ensemble, you know it's the average of H minus T, the Gibbs entropy. Or people who work on more quantum system, it's called the statistical entropy. Yeah. And when I write it explicitly, I use a shorthand notation, which I like usually. I kind of abuse the trace notation. So now with Doppelshock, we now do not quantum. Yeah. So either it's a true trace if you do quantum, it's a phase-space integral if it's some phase-space problem, but in our case, it's simply the sum over omega, just in case, but I think it's an easier notation. But you see also this has a completely generic character. So many arguments I deliver are completely agnostic to the phase separation problem. Okay, so let us write this here. So how's an average defined? We have a density rho, our H, plus we have a sign change when we plug in the entropy, KBT, trace rho, ln rho, okay? So why is this problem hard to solve? I just said, first of all, we have this quadratic interaction here, that's why it's hard. Is it hard because here is an H we can calculate as quadratic, or because here is a quadratic H in the rho? Rho is the Boltzmann factor. So what is the hard step? Is this the hard problem, or is it because rho, in the Boltzmann factor, the quadratic term? The hard term is, it's not that one, because it's just a sigma I, sigma j. And you have all calculated this in the mean field approximation, because by just derivatives, you can pull this in front. That's how you calculate correlation functions. So the hard problem is actually, are these rows here? Okay, how to calculate them? Yeah, so is there a question? No. Was there? No. Okay. So, what to do now? And I can highly recommend Sam Safran's book here, because it's very hard to find. We do define a model Hamiltonian. We call it H naught. It comes with a probability density rho naught, which is 1 over the partition function e to the minus h naught divided by kbt, ugly kbt here. And the partition function is, of course, okay. And we use this model Hamiltonian to replace exactly the problem, and this is exactly the row here. So we define approximate free energy. That is first of all here a talk, and I'll tell you in a second how to choose the model Hamiltonian. Okay, what do I do now? I write the same thing again, but just here with the rho naught. But I keep the h here. That's very important. This I keep. Yeah? Plus kbt trace rho naught logarithm rho naught. Okay? So I've exactly replaced what makes it hard to solve. And then comes a really almost seemingly nonsense equation. One can show that the f approx is always bigger. That's no surprise, right? Because free energy should be minimal. There's one thermodynamic state with minimal free energy. So whatever I do, I get an upper bound. I can rewrite this upper bound with an, yeah, I mean, I think I defined already. I don't need the other form. But for any model Hamiltonian, all right, just shorter. Okay. Okay, so you understand that it's kind of always smaller. And here I wanted to give the reference because Sam Safran, he has a beautiful book that starts with statistical thermodynamics. Of surfaces and membranes, beautiful book. And he nicely described this method there. So, what about this model Hamiltonian? The model Hamiltonian, we have to choose a model Hamiltonian we can deal with, right? And what could that be? What model Hamiltonian of that system can we come up with that we can always deal with? Say it again.
I was trying to understand.
Ah.
I didn't hear the question.
Yeah, okay. Can you even see that from that angle? No, for you it's fine. Okay. Yeah. No problem. So you can use a mean field Hamiltonian, but you have to do a local mean field. So what is this? How to write a local mean field Hamiltonian? We don't have to even think, because mean field is mathematically, can someone say what is mean field mathematically? Physically, you replace the interaction at each site with a mean interaction. Now we do this locally and not globally. But what does it mean mathematically? It corresponds to what? We have sigma I times sigma j. No. In some way, maybe, yeah. Because it kills fluctuations, yeah. What do you do mathematically? You have sigma I, sigma j. That's nonlinear, right? And the mean field Hamiltonian is it nonlinear? it's linear, right? Yeah, so it's a linearization mathematically. So mean field is a, so that physical argument I gave you already, it's a linearization, and I can always linearize. That's a cool thing. Yeah? I can always establish one, but now I write it differently compared to most of your lectures, I think. So this is the linear term that remains. And here now I introduce a field, but I keep the site dependence, okay? I do a local mean field approximation, okay? And that depends, of course, on this local mean field, and we would expect parameter-wise, it also contains here these. So again, exactly, okay. So I can define the, if you want, I can define for you the phi m, this local mean field. Yeah, sorry, I need a bit of here. So I just sum over the sites, and where I is element the neighborhood of M, and here divide, of course, arithmetic mean type also by the amount of next neighbors. Okay, but I do it locally. I do it for each site, for each environment. Okay. Good. Yeah, with this, I think the rho naught we have already here, and this we can now calculate. So now, what's the crux of this mean field Hamiltonian? This local mean field Hamiltonian? We can now, because we know this one, this approximate one, with this local mean field is an upper bound that calls, yeah, when we know it's always bigger, it could be a very bad thing, right? It's just an upper bound, right? It could be just super far off, right? But we could not tune it to bring it closer. So what do we have to do? We have to minimize it. Yeah? That calls for a variational principle. Yeah? So where we can now minimize f approx and vary this local mean field. Yeah? But of course, we, as in all statistical mechanics minimizations, we have constraints. Yeah? And the constraint here is, of course, that the mean composition evaluated with my replaced Hamiltonian, which is just defined as the trace of sigma m rho naught, yeah, is, and this is here really, that's the condition, yeah, is phi m. This phi m. Yeah? Okay. And this you can do now. Yeah? So, because you can do a mean field, there's now a local mean field, and you can do, you know what is the variation principle, you can minimize the function. The rest is algebra. By the way, it's long algebra. It's really, I mean, I did this the airplane when I came here again, and it's also okay to do mistakes. But if you know, of course, what should be the result, you find them easily. But it's really a very nice calculation. And why is it so relevant? Because our f approx, because of course we then say, okay, fine, our f is approximately our f approx. We get it then. And the f approx reads as follows. It reads very similar to this thing that we wrote out down here already. So what do we get? We get here a sum over next neighbor. I don't know, I changed now here to I and j, it seems. I get here 1/2 delta E phi I1 minus phi j. Okay, oh, this thing we have seen here, right? This just went now as a term here into my free energy. And then I get a free energy as units of kBt. I get here a term which is just the sum over all the lattice sides with this local mean field phi m. Okay, that's what you get from this calculation. Yeah, it's a very beautiful thing here. This here, that looks like phi log phi, looks like a mixing entropy, right? Yeah, so if you can play this game, yeah. Have you ever done this? I mean, okay, what is the number of configurations to rearrange two particles on a lattice? Yeah? If you have done this, you get the number of Microstates, this is simply NA over N, was the same as here, this one. You take the logarithm of it, do a Stirling approximation. Okay, that's the Boltzmann entropy, but the Boltzmann entropy is the same as the Gibbs entropy in the thermodynamic limit. And then you get the same term. Okay, so that is as you would have not done a very careful thing. But this term is here very interesting because it contains also spatial heterogeneities. And this I would like to show you now. So now I have to scroll over all my boxes that you can have a look if you want. Because now I would like to do a continuum limit. Good. There's not heavy math, so I just take this term here. I don't know, I wrote now phi n, but sorry, I'm switching a little bit between subscripts, but doesn't matter. Nm is the same as I and j. Yeah? And mathematically, you can just rewrite this term. It's just simple algebra as 1/2 phi n minus phi m squared. And what is left over is a phi n squared minus phi m squared, and then 2 times phi n. Okay, you can just test this. It's just the same. It's just algebra. But why I'm showing this? Because this is now a difference between the lattice sites n and m. And when I have just next neighbor interactions, nothing contributes except me with the interaction with my neighbor and vice versa. So I can down. This is ideal to make a continuum limit. Where I keep here the 1/2, and what do I get? Yeah, I have to come up with a length scale. If it's next neighbor interactions, how to make a gradient? Yeah, just the difference between me and my neighbor, and divided by a length scale. The length scale is A. That's the lattice side, if it's next neighbor interactions. So I have to cook up this length scale here, A squared, gradient phi of x squared. And this one's here, yeah, I just can replace it locally. I don't have to even think. This gives me phi 1 minus phi. Okay, just for clarity, yeah, so here, this is, we have to also take care of the sum over m over the lattice sides. Yeah, so here, sum of lattice sides, and have to introduce here also a continuum integral, and that's how I do it. And the result, what I get, is a very simple free energy. So the total free energy system is the integral over space. There is a free energy density phi plus exactly this gradient term here. That's why we did this extra hard work. There's a bit of length scales here. 1/2 kBt. Sorry. Okay. Oh yeah, I introduced already here a parameter kappa. Okay, so what you see here, I have a free energy, it's integral over space, the density, and I have here a gradient term, now my free energy. Yeah? Who has seen this contribution in your lectures? In your non-equilibrium thermodynamics lectures, maybe? Who has seen that? Yeah? Of course, I mean, there's another way to come up with this, and this is by classics, when people are more interested in critical phenomena. Then they do a Ginzburg-Landau expansion, and then they can just add it. It's a free parameter. Here, it's not the case, because I'm not discussing critical phenomena. I'm discussing discontinuous phase transitions. So the kappa here is not a free parameter. Kappa is now chi, A squared. And chi came from my lattice model. Okay, I haven't defined chi. I'm running out of space. We need more boards. So, but I think I will delete here a little bit the stuff on the left, but I write here at least here the parameters that you have them here on the left-hand side. Kappa is simply my delta E, and now I have to even look it up. You have to divide it by 2 kBt. And also, it gets from the lattice coarse graining. This I did not tell you exactly. But of course, when you sum over my neighbors, you have to then exactly count them correctly. Okay? So whether, now depends on the lattice. To have 4 neighbors, 8 neighbors, and all these kind of things. And this is usually all absorbed into this coordination number of the lattice. And this is exactly what you don't want to have in a continuum theory anyways. Coordination number of lattice, because that is just the artificial aspect of starting with a lattice. Yeah? Okay, and this is, that's why this is really a distinct thing, because the interaction between the molecules, all embedded in this delta E and the chi, determine both. Determine this contribution here, but also determine F. And even though it might look a little bit ugly, I'll write here now the F and then delete the board on the left side, because we can then work with F consistently. Okay? So what is my F? F, how we can all read it well. Yeah. So this is what you get. That's the free energy you've already seen. It's the free energy density, kBt by volume. This is the mixing entropy contribution, and this is our interaction term, chi times phi 1 minus phi. And that's the one I had on the board, yeah, to describe this experiment. And now we derived it from scratch, starting from a simple lattice model. As a short comment, to finish up the link to this experiment, maybe I can quickly go back with the slides. Oh, we would have forward, it was forward, I guess. So you see, so here you see this phase diagram. So it's exactly this free energy which we derived. Almost, there's a weird factor here. I'll come back to this in a second. But here you see the same form here. And with this one, you could fit exactly this phase diagram. The difference is still here we described in terms of temperature. Well, temperature, where's the temperature in the chi? We see here, chi depends on temperature already. It's 1 over temperature. But we are missing something, and this is, we have not accounted, we didn't specify that this one is already maybe a coarse grain type of interaction that contains temperature. Even though it's a microscopic parameter, it can already come from some interaction that has entropic origin. Do you know an interaction where molecules can come together attractively that has an entropic origin? Which one have you seen in your lectures? What do we mean by that? I mean, of course, an enthalpic one would be a glue. Okay, they come together, glue, Lennard-Jones minimum, fine. But there are also cases where the origin is entropic. What do I mean by that? Can someone explain? Ever heard of this? No? Actually, there are many. And I give you one example. For example, when you bring... Oh, I'm running out over time. Oh, okay. Don't give an example. 4 minutes, yeah, okay. Yeah. I will not make it today anyways. So we'll continue then tomorrow. An entropic interaction is, for example, say you think about electrostatics. 2 opposite charge molecules come together. So if they're separated, interact with their counter ions, or come together, the electrostatic energy change is the same. However, what you do is, when you take a molecule with all this counter ion cloud, push the counter ions away, bring the 2 macromolecules together, there's entropic change. This is called counter ion release. It drives conservation and even biomolecular condensation. Very important effect in this type of physics. But there may be many others. Have you heard of depletion effects, for example? This entropic effect, or all these type of polymer physics, there are many kind of cases, such as entropic springs, type of effects polymers can have. These are all of entropic origin. So in principle, the E here depends already here on temperature, and then in polymer physics, since I can't finish anyways, so I add this here, came up with this generic form of A plus B over T. The B over T we have already. Yeah, this is called the enthalpic one, and this is called the entropic one. Okay, and this is exactly what we had used. We also used that the effective molecular volume depends on temperature. This is established knowledge that molecules can extend upon temperature. And then we could fit this phase diagram, a phase diagram in a living cell. I think I find this very striking because in general, one should have expected it shouldn't work due to extremely many reasons. Good, because I have how many minutes left? Just a few. Yeah? I have 20 minutes left. Okay, so I have to clean. Okay, sorry, misunderstanding. I thought you said 4. Okay. Okay. Sure, can read. But how can I clean then the board? Then I can continue because it's just with this one. Just yeah. Yeah, just leave the very right-hand side. I mean, everything, yeah, I think maybe this one I will not continue, but everything from here, yeah, exactly, because, thank you so much, very, very nice. So you have the password, yeah? Good, so the lattice model we have now used to get to this free energy, and now, of course, we want to exploit the effects of the free energy. The thermodynamics part I keep a bit short, because this is very similar to the Ising model anyways. But let us draw this free energy. So for example, I can put the chi to 0, then nothing interesting should happen, so which means there are no interactions, okay? There's just entropy. And I can plot this as a function of this phi between 0 and 1. Then what I get here is a classical convex free energy, and everything is fine. Okay, so convex means that f prime prime of phi is positive. However, when chi is big enough, yeah, so there's one thing I have to maybe mention for generality here for the free energy. So if you do a much harder calculation, you get an extra correction factor, namely this you only get when molecules are of equal size. When nu A is equal, no, delete it. When when they all occupy the same number of lattice sites on the lattice. However, when you correct this, you get here an n factor. Remember, n was simply nu I divided by the molecular volume of the lattice. This accounts for the fact that these are you're not phase separating in general from a colloid of a certain size, from other colloids of the same size. In particular, in this case, I show you, I mean, in the cell, I mean, what is phase separating? Are big proteins, macromolecules, together with other big macromolecules, RNA, from, yeah, you could argue, from other proteins. Actually, it's not so understood, but definitely from water. Yeah? So in general, there's a huge diversity of sizes. Yeah? That's why you have to come up with a free energy model that incorporates also size difference among molecules. And this, among other features, makes the free energy right away asymmetric. So anyone who came up from the Ising model with a symmetric free energy, say goodbye to symmetric free energies because they don't exist in general. Molecules in general have never the same size. Yeah? So the generic case is that they're asymmetric. Yeah? So here, of course, I now drew an almost symmetric one. Amazing. Let's make it a bit like, okay, here's 1/2, okay? So it's somehow asymmetric. Good. Now, if chi is big enough, I cannot tell you now how big it has to be. Yeah? Then the free energy density can look like this. You've all seen this, I suppose, right, in the Ising model. Such a free energy has a problem from the first homogeneous perspective. It has here a concave domain. Which means that the homogeneous state, a homogeneous thermodynamic state, can't be stable there. Do you remember why? Can someone just call in and say why is it not possible that a thermodynamic state can exist in a concave free energy domain? Do you remember? What was the argument in your lecture? So here it's obviously this, what is it, f prime prime, phi is negative. This is concave. What problem does that create? Yeah, it's not a minimal state, exactly. But first of all, from the first one, just look at this. What is the, it's in conflict with what important property? Minimum is already far, it's not really clear, yeah, because you minimize the total free energy. It's not stable, yeah, exactly. And stability, yeah. And stability is always hooked up with a response function. In that case, is the isothermal compressibility. Typically called kappa T, yeah? And this you can actually calculate that this is 1 over f prime. Then you would see, oh, in that domain it's negative, but kappa T is proportional, including the sign, to the variance of the number fluctuations. So this has to be strictly positive, okay? So then this free energy has some, if we did such a hard work and now we create even a problem, but it's not a problem, right? What is the solution to this? Exactly, the Maxwell construction, or better, like convex hull construction. You know what, I need another color. Oh, wow, that's a big one. And the Maxwell construction exactly is it, let's see. I think a nicer word is the convex hull, because you get a convex envelope created, and it comes here with 2 concentrations. So this is, sorry, I've got here the phi, and this is called, we can call this phi 1 and this phi 2. Okay, this is not so nice to write with, so I try to not to do it. Good. Mathematically, we can do an Ansatz. And the Ansatz for phase coexistence. And I'm doing this because we will, this is of course thermodynamic equilibrium or phase equilibrium, but we need this later to define local equilibria at interfaces, to calculate fluxes in out-of-equilibrium scenarios. That's why it's important that everyone is on the same page. So the Ansatz is, no, wait, I called it here for this lecture in and out. I apologize. Okay. Subscript or superscript? Subscript. So you can think about a system like this. I have a box. Even though you have to be careful, not draw a rounded object, but it's fine. We will introduce surface tension and interface in a second. And this is here phase of a, I wrote the other way around, sorry. Out and in. Yeah, so there's an inside of this phase of a higher phi, of a higher macromolecule concentration, and an outside phase, okay? This is the box. It's the box of volume B. Okay, so we can do the Ansatz. The Ansatz is simply phase coexistence, so that the total free energy is, we take the volume of phase 1, multiplied by the density, 1 over volume in terms of units, phi in, plus V out. Oh, a bit unlucky, but fine, yeah. So that's the Ansatz we do, okay? This is the Ansatz of phase coexistence. Later we correct it here. I will here add keep here a box because we are missing here a contribution. Yeah. But let's first slowly do it like this. We have conservation laws. We have here, in general, 4 variables, but because of the conservation laws, we get less. So first of all, we have that, of course, the 2 volumes of the 2 phases, V in plus V out, add up to V. And then we have, of course, that the particle number A inside plus the particle number outside is some sort of total particle number NA, okay? Particles are conserved, yeah? The number of A particles, thus is also the B particle, but this we discarded already from the beginning. And now we can massage this a little bit. You can here multiply and divide by the respective volumes, and yeah, maybe I do it. We can write here V out times nu divided by V out. I can multiply the entire equation by nu. Now this is the molecular volume of the lattice. Yeah. Which means I can write in a nicer way, because now we would like to work with volume fractions for some time. Yeah, so we see very nicely, so the volume fraction outside multiplied by the volume, and the volume fraction inside multiplied by the volume is exactly this conserved variable. And this in the literature has an important name, because I can now solve it, for example, for phi in as a function of V out and phi out, etc., dot, dot, dot. And this is called the lever rule. Yeah. Good. So, if we want to get this Maxwell construction and the equilibrium constraint, we have to minimize the free energy. In principle, I said already we have here 4 unknowns, but we have 2 constraints. Okay, it would be a cumbersome work to introduce now 4 Lagrange multipliers for that, or 2 Lagrange multipliers. So we just plug them in and minimize f as a function of, I can write phi out, but because f is an extensive thermic variable, I better write also the corresponding extensive one, V out. Yeah, so we minimize exactly this function that depends only on 2 variables, which means we get 2 conditions. So the 2 conditions when you minimize are df over dn out is 0, df over v out is 0. Okay? These are the 2 conditions I would like to plug in to 0. So sorry, this is a partial derivative. And so I get 2 conditions. So what do we get? It's not very difficult math, because we have our free energy. Where did I write the free energy? Here it is. So we can now replace everything, right? So the phi in, we can plug in, etc., and then do all these derivatives, and we have a lot of chain rules. I mean, I will not calculate chain rules for you guys, but when you do this, you get that the slope is equal with the first condition, and the second condition gives at first we're different of the free energies. Ooh, sadly I killed my, I have a little bit left, yeah. Should have, but not, yeah, I should have, almost, yeah. The difference of free energies so I get that the slopes of free energy are the same, and I get a condition that makes sure that I have a smooth and continuous function. So the only unique way to do this is exactly the Maxwell construction. It's like whether I have to find function on the free energy where the slope is the same, but there's a continuous change. So for example, this one here, the slope would be, I cannot do this one here, for example. So that's the only way to graphically construct it. And now I'm just sorry that actually here, but I can repair it. I can write here mu bar. This one we don't need. And this is the pi. This is our osmotic pressure, right? Now this we had in the introductory part. Now when you look at this, what is this? F prime. Oh, that's exactly the equivalent of the exchange camp potentials. Outside, inside. And what is this? We have a minus F phi. Oh, we have exactly the same thing. We have here pi out is equal to pi in. So two phases are at equilibrium when the exchange, in the canonical ensemble, when the exchange camp potential and the osmotic pressure are equal between the two phases. Good. So maximum construction we had already. One could calculate, if you want, such a, we can calculate a phase diagram. Analytically, we can't really because, at least for when n, now I have 3 minutes, okay, that's good. I wrap up exactly, perfect, yeah. So let us finish maybe with calculating a phase diagram because now we have the conditions to do it. I quickly show it here for the special case n equal to 1. So this is now the symmetric blend situation. Not blend, symmetric free energy. Yeah, looks like this, symmetric free energy. Okay, it's a bit an artificial case. We have here the same height. Yeah. What we see right away that the osmotic pressure is automatically fulfilled for such a free energy because when we do the construction between the two minima, here the slope is 0 and the free energy difference is the same. Yeah, because here we have the same free energy level of both of the states. Okay, so this is osmotic pressure condition is solved. So in the symmetric case, we're only left with one condition, and this is mu in is mu out is 0. So let's calculate mu bar, which is simply the derivative. So here, that's the formula to calculate it. Here we have the free energy somewhere. Where is it? I can't find it now. Here's it, exactly. So we can calculate simply the phi derivative of this. And when you do this, its camp potential has a scale of kVt, logarithm phi 1 minus phi. What is it? So this is what you get. And when you put this to 0, you can simply solve this for chi. Yeah? And this is called the binodal line. Yeah? And when you do this, you get phi 1 minus phi divided, 1 minus 2 phi. Huh? And this binodal line tells exactly where you phase separate. And another way of saying that, this is called the phase diagram. And it looks like this. In that case, it's symmetric because we made this special choice. Yeah, but as I warned you already, this is not what you want. Inside this domain, you have phase separation. And this we have calculated now from our free energy. So since I'm running out of time, yeah, I did not get as far as I wanted, but which is fine because I just continue maybe in the next lecture and shift everything a little bit. Yeah? But just to quickly summarize what we did, we defined this Hamiltonian, which is pretty similar to the Ising model, but we had these 3 interaction parameters between AA, BB, and AB. Yeah? We had to calculate the free energy. This is notoriously difficult, and I showed you how to do a use a variational principle and a local mean field approximation to get exactly such a nice free energy that also has gradient terms. And we need the gradient terms because they will make up our interface. So if you want to calculate this profile, we need the gradient terms. Okay? And moreover, it defines in the final Canhilla type of equation, which we derive in the next lecture. Now, I finished with calculating phase diagrams. So we just went to the thermodynamic limit of very big phases. We calculated coexistence between phases, and I just drew this without talking about this line of the interface, because we actually did something wrong. We forgot something very important, and this I will continue exactly in the next lecture. We forgot that it costs energy, to build an interface. And the energy associated, so the energy per surface area that free energy you have to pay is the surface tension. And the surface tension will affect the equilibrium concentrations a little bit. That's not why I do it. This I could really skip. But they are very important because they drive phenomena like Oswald ripening, shape relaxation, etc. And that's exactly what we will build on, because later on we will discuss shape instabilities and these kind of things. So thanks for listening. I know it was a little bit, I realized when I asked a bit in the audience, it was maybe a lot for you guys. What I would appreciate if you come to me and ask, then I can more adjust the upcoming lecture tomorrow for your needs. Otherwise, thank you for listening, and I'm ready to take questions.
Questions?
Can you to me again, please? Where did the me equals cavity? This equation, the second equation, yeah.
This one?
Because it looks like those and those, but it's a little bit different.
No, exactly. So what I did, so I used simply that mu bar is nu, and I performed the derivative with respect to phi. So to calculate this one, you just take the f we had here. So this one here. See this?
Yes.
Yeah, this is a function of phi, and you just perform the derivative. And when you plug this in and move the logarithms together, it looks like this. Yeah.
No.
Okay.
Yeah.
You're welcome.
And
Yeah.
again.
Another question?
Yes,
Yeah?
for me. Can you explain again how you did the continuum limit on the side of the board? Because I kind of got lost in all these passages. Yeah.
This one here. Yeah. So after this lattice, of this minimization approach, you get a very simple combination of these local phis here. And it's not so clear here because just phi multiplied by 1 minus phi j. But these are in general different sites. So they contain the information of space as well, if I is unequal to j. And this is exactly what I showed with this simple kind of rewriting, that in actually you can write as a difference squared. Why there's a squared, we can discuss later why there should be a square, because and then you want to build a gradient, but you have to build it on, you have to map it on a scalar thing. So the leading order is gradient squared. So it comes up very naturally, because you cannot, when you have a vector, you have to square it in leading order. This comes very naturally, and these are just local terms. And then when you have this one you see, right? So if you have next neighbor between n and j, say I'm now side 7 and you're side 27, we have no interactions. You're too far away. It's not next neighbor interactions. But say you're the next side, we can build a gradient, so to say, right? And that's only the gradients I account for, because I only make gradients of one lattice side. The A here is arbitrary. So in X-Plane was like, yeah, but you can use another length scale. You can do block spins. You can create blocks. This is what you can do as well. But it doesn't change anything. You have to define a length scale on which you have gradients. And the length scale, the minimal length scale can take is the one of the lattice. Exactly.
Okay, thank you.
Welcome.
Are there more questions?
Hi.
Hey.
I want to ask again if the animation with the droplets and the change in temperature doesn't present an aging in coalescence, because I also think like my friend that it was like a change in the coalescence and absorption, not of the droplets, and it's like very suspicious to me that maybe it's like aging, isn't it?
You mean aging in terms of that the system changes in time to a different state that is with a lower dynamics? You mean aging, right? You said aging, right? Yeah.
That maybe these droplets aren't separating anymore like the first time.
Yeah, but I mean, as you've seen in the movie, they dissolve very nicely, at least after 5 or 10 times change the temperature. So they definitely dissolve, at least what is labeled dissolves, right? Don't see what is left over, maybe. But again, I think you're alluding again to this, what your colleague said, when you look at these volume changes, yeah, that it's not perfectly symmetric. You should not forget this is a living organism composed of 10,000 different type of molecules, undergoing very complex biochemistry. So there's a lot of other things happening in parallel. That even it looks like this, it's reproducible, and it appears like reversible is for me a striking observation. And in the next lecture, I will show you exactly that even this rise of this one here can be nicely explained by the formation of the gradient. Yeah. I'm not saying that this is the only cause, because I mean, this is a cell observation. We only have a limited information, and then we want to make sense of it. But we could just be luck that we give a reason to explain it, but there was something completely else that we did not label, but we did not label exactly 10,000 minus 1 components. This is the challenge, but also the excitement, I guess, of cell biophysics. I would argue that we have limited information, and but still we have to deal with that information to understand mechanisms. Yeah? But one should not oversell the conclusions. So likely we are all wrong in 20 years. I mean, because the system is much more complicated. Yeah? I shouldn't say that, maybe. It's recorded. Okay. Did this help a little bit? Yeah? Yeah?
Yes, perfect, thanks.
Yeah, we can talk also separately after the talk.
It's time to move on to the break.
Yeah, exactly. So thanks a lot.
Thanks again, Christoph.